everyone. My name is Alejandro. I'm a postdoc at Princeton. I'm quite embarrassed to be here for the second time, but thank you so much, the HFSP, for giving me the opportunity to show you a second vignette of the work that I've been doing these years at Princeton, and also for putting together this amazing conference that I'm enjoying a lot. So yeah, today I'm going to tell you still about like microbes, so much simpler than bats or birds. But I'm not going to tell you about like just how they move, and basically not about like phages, like viruses. I'm just going to tell you about like how microbial colonies get their shape in complex environments that mimic their natural environment. So this is actually kind of like the overarching theme of my current research: how living matter like self-organizes and get their shape in complex settings that mimic their natural environments. And the goal is to uncover kind of like these biophysical principles. So today I'll just talk, as I said, about microbial communities, but we have been also looking also in the context of, like, for instance, metabolism, how enzymes can aggregate to maximize yield, and this with also Ned Wingren at the lab of Martin Jonikas at MolBio, and also with 3D tissues, how they respond to for instance, like electrical cues. So as I mentioned, I just focus on microbes. So the central question of the talk is going to be what sets the shape of a bacterial colony. And here I listed like a few reasons of why we care. Like the first one, is fundamentally interesting in biophysics, and basically, bacteria are a canonical model system, but also there are other reasons more applied, like for instance, it's interesting in the context of agriculture. It has impact on the environment, health, and also from a materials perspective, there is a growing interest in developing new forms of soft and living matter, basically. So, of course, there is plenty of previous work, and most of these works, they have focused on characterizing and understanding how bacterial colonies get their shapes when they're growing on 2D planar substrate. So depending on the species, I think this is for Bacillus, but you can build the same for E. coli. You can build these state diagrams in terms of their shape. So here on the y-axis, you have the overall nutrient concentration of the substrate where they're growing in, the agar plate, basically. And on the x-axis, you have the inverse of the agar concentration that tells you about the stiffness. So if you go to the right, basically you have a semi-solid agar plate, like very liquid-like. So they can do what I told you yesterday, like chemotaxis, deplete the nutrients, and then they colonize new terrains where you have more food to consume just by forming these concentric rings. But today, I'm going to tell you more about this other region where the agar concentration is pretty high, so the substrate is stiff. So the microbes, they cannot use their motility, and basically the way that they expand collectively is by consuming nutrients, dividing, growing, dividing, and creating pressure and pushing their progeny outward. And basically here you can find like kind of like two states when nutrients are abundant. You can see that they grow in more like rounded-like shapes, and that makes sense. Like everyone is, nutrients are saturating, so basically, everyone is growing at the same rate, at maximal rate, and you're able to fill in any valley. However, when nutrients are scarce, you grow in more like these, ramified and branch patterns that they are reminiscent of DLA patterns. So, this is great. We have learned a lot from these works, but we, in nature, bacteria, they do not just grow attached on surfaces, but they also, they inhabit like complex 3D spaces. So that was like our main interest. And here, I'm going to just show you like some of examples, like for instance, like the soil, like the ground beneath our feet, which is a complex and granular material. They can also grow as suspended aggregates in large bodies of water, as you can see here, also in bioreactors, and of course in hosts, like for instance in our body. So actually the question that we wanted to understand is what determines the shape of a bacterial colony in 3D. And some of the things that we are now currently doing is what is the interplay between, like these shapes in 3D and, like, for instance, the resistance to a stressor, like antibiotics, phages, or, like, for instance, the genetic diversity, which if I have time, I can show you some, like, recent results that we have. So first, I would like to acknowledge the team. So as I mentioned yesterday, I'm working in the labs of Sujit Datta and Ned Wingren at Princeton. So I'll try to tell you like two stories if I have time. So the first, and feel free to to check the paper. So the first story, the experiments were initiated and led by a former postdoc in the lab, Tapomoy Bhattacharjee, and then Konane, former postdoc, Anna Hancock, grad student, and Hao, former undergrad. They did some additional characterization of the experiments. And in the second part of my talk, if I have time, I will tell you something about like multi-species colonies, which I've been collaborating with Carolina, who is a postdoc in the lab, and the lab of Jeff Gore at MIT, and Hume, who was his grad student there. So basically, in my case, this is how I started the postdoc. I was just supposed to do just theory and simulation, but now this is how it's going. So thank you, the HFSP, for supporting a theorist who wanted to go into the lab and actually being happy there. So yeah, so now the good thing is that I'm much more comfortable explaining the experimental setup because I work with it. So it's the same setup that I just showed you yesterday. We work with these transparent granular hydrogel matrices. So essentially they are made of hydrogen nanoparticles. They are swollen with your favorite media for microbial growth. And basically then you can go with your syringe and go with the needle and inject a pellet of cells of any shape that you would like. You can 3D print anything, and since this is about jamming, then you can remove the needle and it self-heals. So the good thing is that it's transparent, so we can perform confocal microscopy and spy on the cells. As I mentioned, they are easily deformed, they are very squishy, so they do not constrain cellular growth. And they're also replete with oxygen and nutrients, so it's small molecules they can diffuse in, so they sustain cells over long time scales. And then, as I mentioned yesterday, we can tune the degree of confinement. And in particular, today I'm going to focus on the just purely proliferation driven, but before I'm going to show you how it works, just very quickly, so you can see that you can go with a needle and really 3D print any shape that you would like. And this was led by, as I mentioned, the former postdoc in the lab, Tapomoy Bhattacharjee. And you can really, as you saw yesterday, basically we're interested in like just, like 3D printing these kind of like large colonies. So as I mentioned, we can tune the degree of confinement. So for instance, these are E. coli cells, they are motile. However, here we were interested in when they cannot really move. Then we also perform experiments with non-motile cells. But here you can see that the only way that they can collectively navigate this granular material is not by moving, but just by consuming nutrients, growing, dividing, and then pushing their progeny into the next available pore. So that's how they can expand collectively. And we were interested at the colony level, so I'm just going to show you kind of like the main results. So these are under nutrient replete conditions. This is a C projection of maximum fluorescent intensity, and this is just a C projection. So we were expecting from our knowledge in 2D that under nutrient replete condition, they should grow more or less roundedly. However, we observed like something strikingly different. It says they grow, they exhibit this kind of like shape instability, which now they call broccoli-like structure. So you can see the florets, then at small scales, you can see that it roughens. So that was like surprising for us, and then we wanted to understand, we set out to tackle why they do get this shape. So first of all, we started like characterizing them. You can tell just by eye that there is a structure. So the characteristic size or spacing between florets is typically hundreds of microns. And then you can take the leading edge at different angles and compute the power spectrum to see if actually that's true as a function of the wave number. And you can see that most of the power is confined to it plateaus, and most of the power is confined to hundreds of microns in agreement with our visual intuition. And then at a small scale, you see this power law behavior that indicates like a scale invariant, so fractal-like behavior at small scales. And then at the inset, basically, you can see like different replicates under different conditions. So we wanted to understand if this was generic, across species and environmental conditions. So we performed like different experiments just to see if there was something special of being in 3D. So basically, we test this, for like different pore sizes, like matrix deformability, also like nutrient types, like when you have like lots of nutrients or low nutrients, we always observe like the same type of shapes, and even initial colony radius. So at some point, like beyond a critical size, eventually you lose memory and you always adopt like the same type of shape. And we also did like some experiments with other cells, like for instance, Vibrio, that they are biofilm formers. We observed like similar characteristics also with Pseudomonas, and for instance, another microbe with Xylinus sucrofermentans, and we always observe the same. So we thought, okay, maybe this is something intrinsic to 3D, but we wanted to understand why. So we basically inspect a bit more like the experiments from confocal images. So you can take a slice through basically the axis of this kind of like cylinder. And then if you inspect just the signal of GFP, you will see that only cells located at the periphery are metabolically active. That reports on the oxygen level there. So actually, you know, only a thin layer is growing. So that makes sense that maybe, you know, any shape perturbation will be able to be amplified and eventually giving rise to these kind of like florets and rough structures. So then you can do a simple back of the envelope calculation, and then you can compute how much the nutrients can penetrate. So with these plays. And basically, you know, that depends on the diffusivity of either oxygen or your substrate, the uptake rate, and the density, which these are like pretty close back. And these are like tens of microns compared to hundreds of microns of the colony. So that makes sense. And then you can actually ask, like, what are the cells at the core doing? So you can perform experiments with propylium iodide that it's a proxy just for cell death and membrane damage. And you can see that at the core, you have like a lot of like membrane damage, and only cells at these florets are just taking all the nutrients and growing actively. So this is great, but like we wanted to understand, okay, but like what sets the shape? Can we understand like this characteristic length scale and the roughening behavior? So we turned to modeling. So we developed a minimal continuum model that is like coarse graining over many cells. And just borrowing ideas from hydrodynamics. So I'll try to walk you through slowly through the equation. So basically, this is a volume conservation equation. So we treat this as a material that is expanding and growing actively just by consuming nutrients. So we describe it through the local velocity U, and there is injection of volume because there is proliferation. And G is the maximal growth rate of bacteria, and that's modulated by the local availability of nutrients. Then, of course, we need to, since we need to consider momentum conservation. And since we are in a granular material, we decide to take the simplest approach, which is that the velocity is proportional to the pressure gradient, self-induced by proliferation in this case. And then just we have like diffusion and uptake of nutrients and oxygen inside the colony, and diffusion outside. So, you know, with these just minimal ingredients, we wanted to know if this model is able to recapitulate the experiments. So there is something important that is like, eventually there is only, in terms of the physics, there is only one relevant parameter, which is this nutrient penetration length that I mentioned, over the size of the colony, which is a small parameter. And that makes sense. Everything is happening locally, so things should be independent of the geometry. But yet, we wanted to know if this is still able to recapitulate the instability and the roughening. So what we did is just a linear stability analysis. So basically, you impose a shape perturbation, and then you ask if that is going to grow over time or not. So that's this plot here. Like positive means that it's growing over time, that particular wave number or wavelength. Negative remains that it's going to smooth out over time. And these are the colonies growing for different sizes. So you can see as it grows, basically there is a finite range of unstable wavelengths, and with a most amplified wavelength, which is roughly 20 times the nutrient penetration length, that you put like some numbers there that we can measure, is like hundreds of microns, in good agreement with our experimental observation. So basically, you know, differential access to nutrients is driving this instability. So you have a shape perturbation here, like the peaks will have better access to nutrients just due to diffusion, and they will grow faster than the valleys. But of course, at like a small length scales, when you just have like that's driven just by fluctuations, at the same size of these nutrient penetration length, you're also able to fill in the valleys by growing sideways. So that's kind of like the intuition behind. Then you can also run simulations in 3D. You can see that it becomes unstable. I'll go quickly through these. Compare the power spectrum over time. You can see that it plateaus, and we get a reasonable agreement with the experimental power spectrum, and also like the power law behavior. So I hope that I have convinced you that now we understand a bit better. And actually, if you remember, like the snapshot that I showed you at the beginning, they quite resemble how they grow in the lab. So I'll try to tell you very quickly about like a second story, because so far it's going to be quick, I promise. Basically, I've told you that, you know, I've just shown you that results for one particular strain on a species. However, in nature, we know that microbes, they don't live in isolation. In most microbial communities consist of multiple species or heritable phenotypes that they compete for space and nutrients. They can also, you know, cross-feed. So basically, we wanted to understand like the interplay between these kind of like shapes and when you have like different species. And these are just different examples in our body and also in the environment. So this is in collaboration. We decided to take a step back and go to 2D. We're currently addressing the 3D case, and this in collaboration with the lab of Jeff Gore at MIT and with Hyun at MIT and with Carolina, who is a postdoc in Sujit's lab. So basically, Hyun did some experiment with like some soil bacteria, and in some cases, in many cases, he observed that they segregate into this kind of like core cell structure. I won't go into the details about why they segregate, but we were interested like why you can, in some cases, observe like these shapes where you can have a smooth interface outside, like colonizing new terrain, and some smooth interface inside. And in other cases, you find like these kind of like wavy-like behavior. So we decided to try to understand that. And in principle, just purely mechanically, just to see if we can explain it. Then you can go into the literature and classify the experiments in kind of like these two categories. So we developed a similar model. I won't go into details. I'm happy to talk offline. But basically, you have two parameters, like the ratio between proliferation rate between the species and the friction with the substrate. And actually, what we observed, like by running simulations and comparing with the experiments, that typically just differential friction or just differential proliferation rates drive that instability. So when you have a faster proliferator outside, basically you're able to squeeze in like these kind of like fingers and extrude them, which we are now trying to understand why if that's important, what's like the biological implication. Eventually, you can put together a phase diagram, compare with the experiments, and actually you can see that typically, which is the most frequent case, when the outer, the faster proliferator is outside, you will have something that is like morphologically unstable inside, basically. So we are currently trying to extend these ideas to 3D and just to see basically, you know, the interplay between shape and these kind of like, you know, when you have like different species and, you know, the relation with like genetic diversity. I won't have time to go into these. So I'll just leave you with a conclusion. I hope that I have convinced you that, you know, we have a framework for understanding how microbial colonies get their shape. And we are currently also like investigating, you know, how that affects like the resistance for antibiotics and phages. So thank you for your attention, and I'll skip these. And thank you also for the team and the Human Frontier Science. Yeah, happy to take any questions.
Thank you very much. We have time for 2 questions, not more.
On the 3D colony case where you had the analysis showing bumps that develop, is it possible to make a different kind of model where the bumps beget other bumps, which then collide with each other and so forth? Or is that...
Yeah, sure. You can think maybe probably of a kind of like more agent-based type of model, like, you know, tagging like pockets of cells and with different rules. I mean, in 2D, people have done that, so it could be extended to 3D. And in principle, since this is all driven by differential access to nutrients, in principle, they should also observe like the same. But we haven't tried that, but that's a good idea.
Yeah. Thank you for the talk. Your methodology is really interesting, and you have some very cool results. I was wondering, since we know GFP do need O2 for the folding, how do you avoid any possible bias when you measure like really selectivity by GFP expression in possible 3D structure where you may have like availability of O2 that are variable?
I mean, that's why, like, for instance, like we did the PI dye measurement just to see what was going on with the cells at the core, because we weren't sure if like they were just dormant or what was happening. And basically what you observe is that you have like massive membrane damage there, because as I mentioned, like for instance, there can be like also like shadowing issues and other stuff. So that's why we did the PA dye, but that's a good point. Thank you.
Thank you very much. And we